Continuation of the previous video about endocrinology, physiological functions of growth hormone. All the major anterior pituitary hormones except for growth hormone exert their principal effects mainly by stimulating target glands including thyroid gland, adrenal cortex, ovaries, testicles and mammary glands. Growth hormone, however, does not function through a specific target gland but exerts its effects directly on all or almost all tissues of the body. Growth hormone promotes growth of many body tissues. Growth hormone, also called somatotropic hormone or somatotropic, is a small protein molecule that contains 191 amino acids in a single chain and has a molecular weight of 2 to 0.005. It causes growth of almost all tissues of the body that are capable of growing. It promotes increased sizes of the cells and increased mitosis with development of greater number of cells and specific concentration of certain types of cells such as bone growth cells and early muscle cells. Function of growth hormones are important. Growth hormone has several metabolic effects. Aside from its general effect in causing growth hormone has multiple specific catabolic effects including the following, which are the increase rate of protein synthesis in most cells of the body, increased mobilization of fatty acids from adipose tissue, increased free fatty acids in the blood and increased use of fatty acids for energy, and decreased rate of glucose utilization throughout the body. Thus, in effect, growth hormone enhances body protein, decreases fat stores and conserves carbohydrates. Then growth hormone promotes protein deposition in tissues. Although the precise mechanism by which growth hormone increases protein deposition are not fully understood, a series of different effects are known, all of which could lead to enhanced protein deposition. Then enhancement of amino acid transport through the cell membrane. Growth hormone directly enhances transport of most amino acids to the cell membranes to the interior of the cells. This increases the amino acid concentration in the cells and is presumed to be at least partly responsible for the increased protein synthesis. Then enhancement of RNA translation to cause protein synthesis by the ribosomes. Even when the amino acid concentrations are not increased in the cells, growth hormone still increase RNA translation, causing protein to be synthesized in greater amounts by the ribosome in the cytoplasm. Increased nuclear transcription of DNA to form RNA over more prolonged periods. Growth hormone also stimulates transcription of DNA in the nucleus, causing formation of increased quantities of RNA. This promotes more protein synthesis and growth if sufficient energy, amino acids, vitamins and other requisites for growth are available. In the long run, this may be the most important function of growth hormone. Then decreased catabolism of proteins and amino acids. In addition to the increase in protein synthesis, there is a decrease in the breakdown of cell protein. A probable reason for this decrease is that growth hormone also mobilizes large quantities of free fatty acids from the adipose tissue, and these are used to supply most of the energy for the body's cells, thus acting as a potent protein sparer. Summary: growth hormone enhances almost all facets of amino acid uptake and protein synthesis by cells, while at the same time reducing the breakdown of proteins. Then growth hormone enhances fat utilization for energy. Growth hormone has a specific effect in causing release of fatty acids from adipose tissue and therefore increase the concentration of fatty acids in body fluids. Also in tissues throughout the body, growth hormone enhances conversion of fatty acids to acetyl-CoA and its subsequent utilization for energy. Therefore, under the influence of growth hormone, fat is used for energy in preference to use of carbohydrates and proteins. Then ketogenic effect of excessive growth hormone. Under the influence of excessive amounts of growth hormone, fat mobilization from adipose tissue sometimes become too high that the large quantities of acetoacetic acid are formed by the liver and are released into the body fluids, thus causing ketosis. Thus, excessive mobilization of fat from the adipose tissue also frequently causes a fatty liver. Then growth hormone decreases carbohydrate utilization. Growth hormone causes multiple effects that influence carbohydrate metabolism, including decreased glucose uptake in tissues such as skeletal muscle and fat, then increased glucose production by the liver, and increased insulin secretion. Then each of these changes result from growth hormone induced insulin resistance, which attenuates insulin's action to stimulate uptake and utilization of glucose in skeletal muscle and adipose tissue, and inhibit gluconeogenesis, glucose production by the liver. This leads to increased blood glucose concentration and a compensatory increase in insulin secretion. For these reasons, growth hormones effects are called diabetogenetic, and excess secretion of growth hormone can produce metabolic disturbances similar to those found in patients with type 2, that means non-insulin dependent diabetes, who are also resistant to the metabolic effects of insulin. The necessity of insulin and carbohydrate for the growth promoting action of growth hormone. Growth hormone fails to cause growth in animals that lack a pancreas. It also fails to cause growth if carbohydrates are excluded from the diet. The phenomena show that adequate insulin activity and adequate availability of carbohydrates are necessary for growth hormones to be effective. Part of this requirement for carbohydrates and insulin is to provide the energy needed for the metabolism of growth, but there seem to be other effects as well. Especially important is the ability of insulin to enhance transport of some amino acids into cells in the same way that it stimulates glucose transport. Then growth hormone stimulates cartilage and bone growth. Although growth hormone stimulates increased deposition of protein and increased growth in almost all tissues of the body, its most obvious sign is to increase growth of the skeletal frame. This results from multiple effects of growth hormone on bone, including the following: increased deposition of protein by the chondrocytic and osteocytic cells that cause bone growth, then increased rate of reproduction of these cells, and a specific effect of converting chondrocytes into osteogenic cells, thus causing deposition of new bone. Then genic cells, thus causing deposition of new genic. There are two principal mechanisms of bone growth. First, in response to growth hormone stimulation, the long bone growth in length at the epiphyseal cartilage, where the epiphyses at the ends of the bones are separated from the shaft. This growth first causes deposition of new cartilage, followed by its conversion into new bone, thus elongating the shaft and pushing the epiphyses further and further apart. At the same time, the epiphyseal cartilage is progressively used up, so by late adolescence, no additional epiphyseal cartilage remains to provide for further long bone growth. At this time, bony fusion occurs between the shaft and the epiphyses at each end, so no further lengthening of the long bone will occur. Then second, osteoblasts in the bone periosteum and in some bone cavities deposit new bone on the surface of older bone. Simultaneously, osteoclasts in the bone remove old bone. When the rate of deposition is greater than that of restoration, thickness of the bone increase. Growth hormone strongly stimulates osteoblasts. Therefore, the bones will continue to become thicker throughout life under the influence of growth hormone. It is especially true for the membranous bones. For instance, the jaw bones can be stimulated to grow even after adolescence, causing forward protrusion of the chin and lower teeth. Otherwise, the bones of the skull can grow in thickness and give rise to bony protrusions over the eyes. Then, growth hormone exerts much of its effect through intermediate substances called somatomedians. When growth hormone is applied directly to cartilage chondrocyte culture outside the body, proliferation or enlargement of the chondrocytes usually fails to occur. Yet, growth hormone injected into the intact animal does cause proliferation and growth of the same cells. In brief, growth hormone causes the liver and, to a much less extent, other tissues to form several small proteins called somatomedians that have the potent effect of increasing all aspects of bone marrow growth. Many of the somatomedian effects in growth are similar to the effects of insulin on growth. Therefore, the somatomedians are also called insulin-like growth factors or IGFs. At least 4 somatomedians have been isolated, but by far the most important of this is somatotropin somatomedin C, also called insulin like growth factor 1 or IGF-1. The molecular weight of somatomedin C is about 7,500 and its concentration in the plasma closely follows the rate of growth hormone secretion. The pygmies of Africa have congenital inability to synthesize significant amount of somatomedin C. Therefore, even though their plasma concentration of growth hormone is either normal or high, they have diminished amounts of somatomedin C in the plasma, which apparently accounts for the small stature of this people. Some other drafts also have this problem. It has been postulated that most, if not all, of the growth effects of growth hormone result from somatomedin C and other somatomedins rather than from direct effects of growth hormone on the bones and other peripheral tissues. Some aspects of somatomedin hypothesis is still questionable. One possibility is that growth hormone can cause the formation of enough somatomedin C in the local tissue to cause local growth. It is also possible that growth hormone is directly responsible for increased growth in some tissues and that the somatomedin mechanism is an alternative means of increasing growth but not always a necessary one. Then short duration action of growth hormone but prolonged action of somatomedin C. Growth hormone attaches only weakly to the plasma proteins in blood. Therefore, it is released from the blood into the tissue rapidly, having a half-life in blood of less than 20 minutes. By contrast, somatomedin C attaches strongly to a carrier protein in the blood that, like somatomedin C, is produced in response to growth hormone. As a result, somatomedin C is released only slowly from the blood to the tissues with a half time of about 20 hours. This slow release greatly prolongs the growth promoting effects of the bursts of growth hormone secretion. Then regulation of growth hormone secretion after adolescence. Secretion decreases slowly with aging, finally falling to about 25% of the adolescent level in very old age. Growth hormone is secreted in a pulsatile pattern, increasing and decreasing. The precise mechanisms that control secretion of growth hormone are not fully understood, but several factors related to person's state of nutrition or stress are known to stimulate secretion, such as starvation, especially with severe protein deficiency, and hypoglycemia or low concentration of fatty acids in the blood, exercise, excitement, trauma, and ghrelin, a hormone secreted by the stomach before meals. Then growth hormone also characteristically increases during the first 2 hours of deep sleep. The normal concentration of growth hormone in the plasma of an adult is between 1.6 and 3 mg per ml. In a child of adolescent, it is about 6 mg per ml. These values may increase to as high as 50 mg per ml after depletion of the body stores of proteins or carbohydrates during prolonged starvation. Under acute conditions, hypoglycemia is a far more potent stimulator of growth hormone secretion than in an acute decrease in protein intake. So conversely, in chronic conditions, growth hormone secretion seems to correlate more with the degree of cellular protein depletion than with the degree of glucose insufficiency. For instance, the extremely high levels of growth hormone that occur during starvation are closely related to the amount of protein depletion. This demonstrates the effect of protein deficiency or kwashiorkor on plasma growth hormone, and then the effect of adding protein to the diet. These results demonstrate that under severe conditions of protein malnutrition, adequate calories alone are not sufficient to correct the excess production of growth hormone. The protein deficiency must also be corrected before the growth hormone concentration will return to normal. Then role of the hypothalamus, growth hormone releasing hormone, and somatostatin in controlling growth hormone secretion. It is known that growth hormone secretion is controlled by two factors secreted in the hypothalamus and then transported to the anterior pituitary gland through the hypothalamic hypophyseal portal vessels. They are growth hormone releasing hormone and also called as somatostatin. Both of these are polypeptides. GHRH is composed of 44 amino acids and somatostatin is composed of 14 amino acids. The part of the hypothalamus that causes secretion of DHRH is the ventromedial nucleus, which is also sensitive to blood glucose concentration, causing safety in hyperglycemic states and hunger in hypoglycemic states. The secretion of somatostatin is controlled by other nearby areas of hypothalamus. Therefore, it is responsible to expect that some signals that modify a person's behavior feeding insects also alter the rate of growth hormone secretion. In a similar manner, hypothalamic signals depicting emotion, stress, and trauma can all affect hypothalamic control of that catecholamines, dopamine, and serotonin, each of which is released by a different neuronal system in the hypothalamus, all increase the rate of growth hormone secretion. When growth hormone is administered directly into the blood of an animal over several hours, the rate of endogenous growth hormone secretion decreases. This decrease demonstrates that growth hormone secretion is subject to typical negative feedback control, as is true for potentially all hormones. The nature of this feedback mechanism and whether it is mediated mainly through inhibition of GHRH or enhancement of somatostatin, which inhibits growth hormone secretion, are uncertain. Then abnormalities of growth hormone secretion. Panhypopituitarism. Panhypopituitarism means decreased secretion of all the anterior pituitary hormones. The decrease in secretion may be congenital, present from birth, or it may occur suddenly or slowly at any time during life, most often resulting from a pituitary tumor that destroys the pituitary gland. Then panhypopituitarism in the adult. Panhypopituitarism first occur in adulthood, frequently results from one of three common abnormalities. Craniopharyngiomas, a tumorous condition involving the pituitary gland. Then chromophobe tumors may compress the pituitary gland until the functioning anterior pituitary cells are totally or almost totally destroyed. Then thrombosis of the pituitary blood vessels. This abnormality occasionally occurs when a new mother develops circulatory shock after the birth of her baby. The general effects of adult hypopituitarism are hypothyroidism, depressed production of glycocorticoids by the adrenal glands, and suppressed secretion of gonadotropic hormones, so that sexual functions are lost. Thus, the picture is that a lethargic person from lack of thyroid hormones who is gaining weight because of lack of fat metabolization by growth adenocorticotropic, adenocortical, or thyroid, and has lost all sexual functions. Except for the abnormal sexual functions, the patient can usually be treated satisfactorily by administrating adenocortical and thyroid hormones. Dwarfism. Most instances of dwarfism result from general deficiency of anterior pituitary secretion, panhypopituitarism, during childhood. In general, all the physical part of body develop in appropriate proportion to one another, but the rate of development is greatly decreased. A person with panhypopituitary dwarfism does not pass through the puberty and never secretes sufficient quantities of gonadotropic hormones to develop adult sexual functions. In one-third of such dwarfs, however, only growth hormone is deficient. These persons do mature sexually and occasionally reproduce. The one type of dwarfism, the African pygmy and the Levi-Lourian dwarf, the rate of growth hormone secretion is normal or high, but there is a hereditary inability to form somatomedincy, which is a key step for the promotion of growth by growth hormone. Then treatment with human growth hormone. Growth hormones from different species of animals are sufficiently different from one another, and they will cause growth only in one species or at most closely related species. For this reason, growth hormone prepared from lower animals, except to some extent some primates, is not effective in human beings. Therefore, the growth hormones of the human being is called HGH to distinguish it from others. Then HGH is synthesized by Escherichia coli bacteria as a result of successful application of recombinant DNA technology and is available in sufficient quantities for treatment purpose. Dwarfs who have pure growth hormone deficiency can be completely cured if treated early in life. HGH may also proved to be beneficial in other metabolic disorders because of its widespread metabolic functions. In gigantism, occasionally the acidophilic growth hormone producing cells of the anterior pituitary gland become excessively active, and sometimes even acidophilic tumors occur in the gland. As a result, large quantities of growth hormones are produced. All body tissues grow rapidly, including the bones. If the condition occurs before adolescence, before the epiphyses of long bones have become fused with the shafts, height increases so that the person becomes a giant up to 8 feet tall. The giant ordinarily has hyperglycemia, and the beta cells of the islands of Langerhans in the pancreas are prone to degenerate because they become overactive owing to the hyperglycemia. Consequently, in about 10% of giants, full-blown diabetes mellitus eventually develops. In most giants, panhypothyroidism eventually develops if they remain untreated, because the gigantism is usually caused by tumor of the pituitary gland that grows until the gland is destroyed. The eventual general deficiency of pituitary hormones usually causes death in early adulthood. However, once gigantism is diagnosed, further effects can often be blocked by microsurgical removal of the tumor or by irradiation of the pituitary gland. Then acromegaly, if an acidophilic tumor occurs after adolescence, that is after the epiphyses of the long bones have fused with the shafts, the person cannot grow taller, but the bones can become thicker and the soft tissue can continue to grow. This condition is known as acromegaly. Clinical features of gigantism: height increase up to 8 feet tall, large hands and feet, soft tissues grow rapidly, hyperglycemia usually present leading to full-blown diabetes mellitus. All features of panhypopituitarism if the cause is due to a tumor: weight gain, loss of sexual functions, decreased immunity. Features of hypothyroidism: features of adenocortical insufficiency. Then clinical features of acromegaly: marked enlargement in the bones of the hands and feet, enlargement of membranous bones, rhenium, nose, bosses of the forehead, supraorbital ridges become prominent, protrusion of lower jaw, that means prognathism, protrusion of the vertebrae increase in size, enlargement of soft tissue organs, tongue become greatly enlarged, liver become greatly enlarged, kidney become greatly enlarged. Next topic, posterior pituitary gland. Our learning objectives include: list the actions and factors controlling the secretion of oxytocin and vasopressin, list the functions of oxytocin, list the functions of ADH or vasopressin, then describe the disorders of posterior pituitary hormone secretion, which will be dealt with this video or with the help of next video. So let's start. Posterior pituitary hormones synthesized by cell bodies in the hypothalamus. The bodies of the cells that secrete the posterior pituitary hormones are not located in the pituitary gland but are large neurons called magnocellular neurons located in the supraoptic and paraventricular nuclei of the hypothalamus. The hormones are then transported in the axoplasm of the neuron's nerve fiber, passing from hypothalamus to the posterior pituitary gland. And posterior pituitary gland and its relation to the hypothalamus. The posterior pituitary gland, also called neurohypophysis, is composed mainly of glial-like cells called pituocytes. The pituocytes do not secrete hormones; they act simply as a supporting structure for a large number of terminal nerve fibers and germinal nerve endings from nerve tracts that originate in the supraoptic and paraventricular nuclei of the hypothalamus. These tracts pass to the neurohypophysis through the pituitary stalk or hypophyseal stalk. The nerve endings are bulbous knobs that contain many secretory granules. These endings lie on the surface of capillaries where they secrete two posterior pituitary hormones: ADH, also called vasopressin, and oxytocin. If the pituitary stalk is cut above the pituitary gland but the entire hypothalamus is left intact, the posterior pituitary hormones continue to be secreted normally. After transient decrease for a few days, they are then secreted by the cut ends of the fibers within the hypothalamus and not by the nerve endings in the posterior pituitary. The reason for this is that the hormones are initially synthesized in the cell bodies of the supraoptic and paraventricular nuclei and are then transported in combination with the carrier proteins called neurofisins down to the nerve endings in the posterior pituitary gland, requiring several days to reach the gland. Then ADH is formed primarily in the supraoptic nuclei. Oxytocin is formed primarily in the paraventricular nuclei. Then each of these nuclei can synthesize about 1/6 as much of the second hormone as of its primary hormone. When nerve impulses are transmitted downward along the fibers from the supraoptic or paraventricular nuclei, the hormones is immediately released from the secretory granules in the nerve endings by the usual secretory mechanism of exocytosis and is absorbed into adjacent capillaries. Both the neurohypophysin and hormones are secreted together, but because they are not loosely bound to each other, the hormone separates almost immediately. The neurophysin has no known function after leaving the nerve terminals. Each of these nuclei can synthesize about 1/6 as much of the second hormone as of its primary hormone. When nerve impulses are transmitted downward along the fibers from the supraoptic or paraventricular nuclei, the hormone is immediately released from the secretory granules in nerve endings by the usual secretory mechanism of exocytosis and is absorbed into adjacent capillaries. Both the neurophysin and the hormones are secreted together, but because they are only loosely bound to each other, the hormone separates almost immediately. The neurophysin has no known function after leaving the nerve terminals. Then, antidiuretic hormone. Physiological function of antidiuretic hormone. The injection of extremely minute quantities of ADH as small as 2 mg can cause decreased excretion of water by the kidneys, antidiuresis. Briefly, in the absence of ADH, the collecting tubules and ducts become almost impermeable to water, which prevents significant reabsorption of water and therefore allows extreme loss of water into the urine, also causing extreme dilution of the urine. Conversely, in the presence of ADH, the permeability of the collecting ducts and tubules to water increase greatly and allows most of the water to be reabsorbed as the tubular fluid passes through these ducts, thereby conserving water in the body and producing very concentrated urine. Without ADH, the luminal membrane of the tubular epithelial cells of the collecting ducts are almost impermeable to water. Okay, impermeable to water. And then all the events described previously occur within the 5 to 10 minutes. The aforementioned flow chart, which is something like that, mechanism of action of ADH. Tubular epithelial cells with special vesicles that have highly water permeable pores called aquaporins. ADH combines with the membrane receptors and activates the formation of CAMP inside the cell. This causes phosphorylation of elements in the special vesicles. The vesicles then insert into the apical cell membranes, thus providing many areas of high water permeability. Then, in the absence of ADH, the entire process reverses in another 5 to 10 minutes. This process temporarily provides many new pores that allow free diffusion of cell water from the tubular fluid through the tubular epithelial cells and into the renal interstitial fluid. So, water is then absorbed from the collecting tubules and ducts by osmosis in relation to the urine concentrating mechanism of the kidneys. So, regulation of antidiuretic hormone production. Increased extracellular fluid osmolarity stimulates ADH secretion. When a concentrated electrolyte solution is injected into the artery that supplies the hypothalamus, the ADH neurons in the supraoptic and paraventricular nuclei immediately transmit impulses into the posterior pituitary to release large quantities of ADH secretion to as high as 20 times normal. So, conversely, injection of a dilute solution into its artery causes cessation of the impulses and therefore almost total cessation of ADH secretion. Thus, the concentration of ADH in the blood fluids can change from small amounts to large amounts or vice versa if only a few minutes. Somewhere in or near the hypothalamus are modified neuron receptors called osmoreceptors. When the extracellular fluid becomes too condensed, fluid is pulled by osmosis out of the osmoreceptor cell, decreasing its size and initiating appropriate nerve signals in the hypothalamus to cause additional immediate secretion. Conversely, when the extracellular fluid becomes too dilute, water moves by osmosis in the opposite direction into the cell, which decreases the signal for immediate secretion.